2023. To outline today's event, the webinar in its entirety will run approximately 2.5 hours, which includes a discussion panel and a Q&A session. As a reminder, if you have questions now or during the webinar, please enter those questions into the Q&A chat pod. We'll answer as many questions as time allows. Our speakers for today's webinar includes Dr. Lei Zhang, Deputy Director, Office of Research and Standards and Office of Generic Drugs, Dr. Nilufer Tampal, Associate Director for Scientific Quality, Office of Bioequivalence and OGD, and Dr. M.J. Kim, Director, Division of Therapeutic Performance 2 and ORS within OGD. Please join me to welcome our first presenter, Dr. Zhang.
Thank you for the introduction. Good afternoon. My name is Lei Zhang. I'm the Deputy Director of the Office of Research and Standards, ORS, in Office of Generic Drugs, OGD, at FDA. Today, along with my colleagues, Dr. Nilufer Tampal, Associate Director for Scientific Quality from the Office of Bioequivalence in OGD, and Dr. Myong-Jin Kim, Director of the Division of Therapeutic Performance 2 in ORS OGD, we will present to you this webinar on navigating the first generic drug draft guideline, M13A Bioequivalence for immediate release solid oral dosage forms. This draft guidance was developed by the International Council for Harmonization of Technical Requirements for Pharmaceutics for Human Use, ICH, M13 Expert Working Group, EWG, and was endorsed by the ICH Assembly in December 2022. I'm the rapporteur of the M13 EWG. For M13A, Dr. Tampal served as the topic lead for FDA, and Dr. Kim served as the deputy topic lead. FDA published this draft guidance along with a Federal Register notice to solicit public comments in January 2023. In today's webinar, we will go over objectives of the webinar, provide an overview of ICH M13 guideline series, discuss highlights of M13A draft guideline with an overview of the content, discuss major differences between draft M13A and FDA's current draft ANDA-B guidance, and provide additional discussion on selected topics. We will end the presentation with a summary. Following the presentation, there will be a panel discussion with additional panelists from the FDA. Finally, we will address audience questions at the Q&A session. The main objectives of today's webinar are to provide an overview of the draft M13A guidance, explain ICH Expert Working Group's current scientific thinking on selected topics, and provide clarification on the FDA planning on the implementation of M13A for generic drug applications. Although M13A covers both generic and new drugs, today's webinar's focus is on generic drugs. Generic drugs comprise a significant portion of the pharmaceutical market. In the U.S., generics represent 90% of prescription filled, yet account for only 18% of prescription drug spending. Generic drugs are often the product of a global supply chain and are produced with the intent to market in multiple jurisdictions. BE assessment is important for establishing therapeutic equivalence for generic drug products to their respective comparator products. Common standards for global development for generics can improve access to generics medications. In November 2018, ICH endorsed a reflection paper on further opportunities for harmonization of standards for generic drugs that outlines a strategic approach for developing and enhancing ICH guidelines to support the harmonization of scientific and technical standards for generic drugs, starting from non-complex products, then move to more complex products. Following in the endorsement of the ICH reflection paper in 2018, the Generic Drug Discussion Group, GDG, was formed in 2019 under ICH to serve as a technical discussion group for issues relevant to harmonization of scientific and technical standards for generic drugs. The GDG recommended areas for harmonization under ICH and assessed feasibility of harmonization of various topic areas within existing regional regulatory frameworks. They proposed to harmonize BE standards for immediate release solid oral dosage form as the first ICH guideline to harmonize BE standards for generic drugs under ICH. This proposal was endorsed by ICH in 2019. Subsequently, the M13 Expert Working Group was formed in 2020 to work on a series of ICH M13 guidelines. The objective of M13 are to focus on immediate release solid oral dosage forms designed to deliver drugs to the systemic circulation, for example, tablets, capsules, granules, powders for oral suspension. Also provide recommendations on conducting bioequivalence studies during product development and post-approval phases. Furthermore, to harmonize current regional guidelines or guidances, therefore to reduce the need to conduct multiple BE studies for multiple jurisdictions. And finally, support streamlined global drug development. M13 guideline development includes 3 tiers with their respective scopes. Tier 1 topics are included in M13A, which is the first guideline in the series. It includes scientific and technical aspects of study design and data analysis to support BE assessment. How regulatory decisions are made based on the BE assessment is out of scope. It is acknowledged that the acceptance of comparator products across regulatory jurisdictions could reduce burden of multiple clinical trials. However, it is governed by local laws, therefore it is out of scope. Tier 2 topic, which is BE for additional strength, including biowaiver considerations, will be covered under M13B, which is the second guideline in the series. The relationship to the biostrength, which the BE study is conducted, and also biowaiver from requirement for additional studies will be discussed in this guidance, with the consideration such as dose proportionality in the pharmacokinetics of the comparator product, qualitative and quantitative composition comparison among strengths, comparative in vitro dissolution, and also assessment of the similarity between the dissolution profiles will be used to support a biowaiver for additional strength. Tier 3 topics will be included in M13C, which is the third guideline in the series. It will cover BE study design, analysis, and assessment for highly variable drugs, drugs with narrow therapeutic index, and complex BE study design and analysis considerations, for example, adaptive BE study design. These 3 guidelines will be developed in sequence following the ICH process of harmonization, which includes 5 steps. Step 1 is consensus building on the technical document. Step 2 is the endorsement of the technical document by ICH parties and adoption of the draft guideline by regulators. Step 3 is regulatory consultation and discussion to finalize the draft guideline. Step 4 is adoption of the final guideline. And Step 5 is the implementation of the final harmonized guideline. Development of M13A started in July 2020 and reached Step 2 in December 2022. It is currently in Step 3 of public consultation. The M13 Expert Working Group started to work on M13B in November 2022, and it is currently ongoing while waiting for public comments on M13A. The group with the goal to reach Step 1 for M13B in November 2023. The EWG plans to start M13C after M13B reached Step 2. Now let's turn our attention to M13A. M13A is the first guideline in the M13 series that was developed following the ICH refreshing paper on further opportunities for harmonization of standards for generic drugs. It is currently at Step 3 for public consultation. All 10 regulatory members of the M13 EWG have published the M13A in their regions to collect comments. The deadlines are between April and May. FDA's deadline for comments was on April 3rd. You may still submit comments to some other regulatory agencies with a later deadline. Comments received will be reviewed by the EWG when finalizing M13A. M13A covers the scientific and technical aspect of study design and data analysis to support BE assessment for orally administered IR solid oral dosage forms. It has 5 sections. Section 1 is the introduction that discusses objective, background, and scope of the guidance. Section 2 covers general principles in establishing BE, with 2 main subsections that discusses study design for PK endpoint BE studies and data analysis for non-replicate study design. The replicate study design and other more complicated data analysis will be covered in M13C. Section 3 covers specific topics with 4 subsections. They are endogenous compounds, other immediate release dosage forms, fixed dose combination, and pH dependency. Other IR dosage forms include orally disintegrated tablets, chewable tablets, and oral suspensions. Section 4 discusses documentation, and Section 5 includes glossary. Next, I will go over the M13A content with a little bit more details. M13A covers both generic drugs and new drugs. BE assessment is important for establishing therapeutic equivalence for generic drugs to their respective comparator products. For new drugs, there may be situations when demonstration of BE may be critical for approval decisions. Additionally, BE studies are used by both new and generic drugs for supporting post-approval formulation and/or manufacturing process changes. The guidance further talks about that the PK principles are generally discussing M13A are generally applicable to non-orally administered products with immediate action in which reliance on systemic exposure measure is suitable for establishing BE, for example, certain rectal inhalation and nasal drug products. Deviations from the recommendation in this guideline may be acceptable if appropriate scientific justification is provided. Applicants are encouraged to consult the regulatory authorities when an alternative approach is proposed or taken. M13A refers to ICH M9 Biopharmaceutics Classification System, or BCS-based biowaiver for oral IR products, and it also refers to ICH E6 Good Clinical Practice for Principles and Recommendation for Conduct B Studies in Human Subjects. It further discussed that the following topics are out of scope. That is, how regulatory decisions may be made based on BE assessment, acceptance of comparator products across regions, BE for additional strength of IR products, BE for highly variable drugs and NTI drugs, and also PK study design or data analysis to support bioavailability assessment for new drugs are out of the scope. As I mentioned earlier, BE for additional strength will be covered in M13B, and BE for highly variable drugs and NTI drugs will be covered in M13C. The guidance recommends B study to be conducted in healthy subjects with a randomized single-dose 2-period 2-sequence crossover study design. The guidance further delineates that for safety or tolerability reasons, the B studies may be conducted either in a single-dose or multiple-dose study in patients. Furthermore, for drugs with a long elimination half-life, a parallel study design may be acceptable. The guidance also discusses about the consideration for the sample size calculation for B studies. Test product used in the B studies should be representative of product to be marketed. Comparator product is a terminology introduced in M13A to define the product accepted by regulatory authorities that can be compared against the test product in a B study. In addition to 2-arm, 1 test, 1 comparator study design, the guidance also discusses about B studies with multiple comparator products or multiple test formulations. These multiple-arm B study design could help reduce the need to conduct multiple 2-arm B studies and improve efficiency and streamline the global drug development. These red stars are added to indicate the topics that are either different from the current FDA NDA-B guidance or not discussed in the current FDA NDA-B guidance. They will be discussed in more details later in the webinar. Currently, the recommendation on B studies with regard to fasting and/or fed conditions is different among different jurisdictions. To harmonize, M13A EWG proposed a risk-based approach for determining whether BE should be conducted under fasting, fed, or both conditions, and the meal types for fed B studies. The selection of the B study condition will be based on the dosing instructions of the comparator product, the properties of the drug substance, and the properties of the products being compared. The B recommendations will be different between a non-high risk and a high-risk product. High-risk product is a new concept delineated in M13A and is not currently in any of the regional guidelines. M13A defines high-risk product as the product where the complexity of the formulation design or manufacturing process leads to an increased likelihood that in vivo performance will be impacted differently by varying gastrointestinal conditions between the fasting and the fed states. Here, the risk refers to the risk of B failure. If B is only conducted under one condition, for example, fasting condition, which may not be able to ensure the BE in the fed conditions. BE recommendations with regard to meals are different for high-risk and non-high-risk products. That will be covered in more details later in the webinar. For products with multiple strengths, the strength to be studied in the BE study is dependent on the pK and solubility of the drug. In general, if pK is dose proportional, the highest strength should be studied in BE. If pK is not dose proportional, based on what's known on the reason of the nonlinearity, either the highest strength, the lowest strength, or both strengths should be studied to select the sensitive dose. Additional strength biowaiver will be covered in M13B. In terms of moieties to be measured for B assessment, typically parent and a non-achiral assay for racemate are used for B assessment. The guidance further delineate when the metabolite should be measured and when the enantiomer assay may be necessary for B assessment. For sampling, sufficient samples should be collected, including the pre-dose sample, to obtain a reliable estimation of the absorption phase, the Tmax, and the extent of exposure. AUC 0 to T should cover at least 80% of the AUC 0 to infinity. The guidance further discussed about how to avoid the first point Cmax and use of truncated AUC for long half-life drugs, and also use of partial AUC that may be capture the early exposure of the IR product that may require early onset. The primary PK parameters recommended by M13A include AUC 0 to T or truncated AUC, Cmax, and partial AUC where applicable. Additional parameters such as AUC 0 to infinity may be used to help assess the acceptability of the B study. AUC 0 to T should cover at least 80% of AUC 0 to infinity. The guidance also recommends the standard statistical approach to be employed for the non-replicate BE studies. And BE can be concluded if the 90% confidence interval for the geometric mean ratio of the primary PK parameters lie within a range of 80 to 125%. Specific topic session covers the BE assessment for drugs that are endogenous compounds, other IR dosage forms including ODTs, chewable tablets, and oral suspensions, BE considerations for fixed dose combination FDC products, where the BE should be demonstrated for all components in the FDC according to the principles described in the guideline for non-fixed dose products. Finally, the pH dependency session would cover the BE study considerations for drugs with pH-dependent solubility, where its absorption may be affected by the gastric pH. It will be discussed in more details later in the webinar. The documentation session of the M13A discussed the high-level expectation on the report of the BE study in the submission. The report of BE study should include the complete documentation of its protocol, conduct, and evaluation. Full study reports should be provided for the BE studies upon which the applicant relies for approval. The guidance further refers to ICH E3, structure and the content of clinical study reports for additional details. With that, I conclude the presentation on the overview of M13A content. Now I will turn over to Dr. Tampal, who will discuss the major differences between the draft M13A and the current FDA's draft ANDA BE guidance.
Lei, thank you for setting the stage for us. Hello, everyone. We heard Dr. Zhang provide an excellent overview of the guideline and its scope, and highlight the major differences between the draft M13A and FDA's bioequivalence guidance with PK endpoints for ANDAs. Now we will transition to talk about the recommendations in M13A for select topics. These topics are either the ones that are newly introduced in M13A and not described in the FDA's PK endpoint guidance for ANDAs, or they differ in the recommendations from those in the FDA's guidance. Some of these differences will present a change from the FDA's current practices when the ICH guideline is finalized and adopted by the FDA. And that would be at step 5 of the ICH guideline development process. The first major change I'd like to cover is in section 2.1.5 of M13A, fasting and fed conditions. This section introduces multiple concepts which differ from FDA's current thinking related to the study conditions to be considered for bioequivalence testing. First, the section introduces the concept of high-risk and non-high-risk IR products. Second, it describes situations where testing under both fasting and fed conditions may be needed, and other times where a single bioequivalence study under fasting or fed conditions will suffice. Thirdly, it introduces options for selection of the meal type according to the risk category. This is in contrast to FDA's recommendations for using high-fat, high-calorie standardized meal for all IR products. The terms high risk and low risk are referring to the level of the risk for bioequivalence failure. M13A refers to high-risk products as drug products for which in vivo performance is likely to be impacted differentially by the GI conditions between the fasting and the fed states. So for such products, the results from a fasting study cannot be extrapolated to predict the fed BE outcome and vice versa. Then, BE studies would need to be conducted under both fasting and fed conditions. High-risk products will generally have complex formulations and/or manufacturing processes. In most other cases, for IR products, the results from a fasting BE study may be extrapolated to predict the fed BE outcome and vice versa. In those cases, bioequivalence testing under only one condition, which is generally the fasting condition, is sufficient. M13A refers to such products as non-high-risk products. This slide shows the differences in the recommendations between the FDA guidance and M13A as it relates to the fasting and fed conditions. FDA's current practice is to recommend fasting for bioequivalence under both fasting and fed conditions for all IR products, without considering the dosing recommendations with respect to food. And exceptions to this general practice are based on safety considerations. For example, FDA asks for a fed study only when serious adverse events are anticipated under fasting conditions. Similarly, a fasting BE study only is recommended if the products are clearly labeled to be taken on an empty stomach, or SAEs, the serious adverse events, are anticipated under fed conditions. The ICH guideline, on the other hand, makes a distinction for the testing conditions based on dosing instructions in the labeling, as well as the properties of the drug substance and product formulation. For high-risk products, as mentioned earlier, the guideline recommends to test for bioequivalence under both fasting and fed conditions, irrespective of the dosing recommendations in the labeling, except, of course, for safety concerns. For non-high-risk products, a fasting-only study is recommended except when serious adverse events are anticipated under fasting, in which case a fed-only study is recommended. A fed-only study is also recommended when the product is labeled to be taken with food for PK reasons. For example, increased absorption or reduced variability, or when serious adverse events are expected in fasting condition. And then lastly, either a fasting or fed bioequivalence would be acceptable, where the labeling indicates intake only under fed conditions due to tolerability reasons, such as stomach irritation. Now, to further clarify the high-risk products, M13A mentions examples of drug products that are likely to have a differential formulation-dependent impact on the drug absorption under fasting and fed conditions. These generally include products with low solubility drug substances that are formulated using specialized technologies and/or manufacturing methods to enhance the solubility or to optimize the food effect. For example, microemulsions, amorphous solid dispersions, lipid-based formulations, and nanotechnology. If only one, either the test or the reference product is formulated using this type of manufacturing technologies, or if the test or the reference differs in the particle size control method, for example, the active of the test may be micronized, but of the reference may not be, then the concerns for the formulation-dependent impact on the drug absorption would still exist, and bioequivalence under fasting and fed conditions is recommended. Also, products with differences in excipients that are likely to impact the dissolution, the solubility, or the permeability of the test and reference comparator product differently may also present a risk for bioequivalence failure under the conditions of the GI conditions of the fasted and fed states. For selection of the meal type, for high-risk products, irrespective of the description in the product labeling with respect to the food intake, the fed BE study should be conducted using high-fat, high-calorie meal. This meal has the potential to cause the greatest effect on the GI physiology. We want to test under the most stringent condition. And this recommendation also aligns with FDA's current recommendation for the high-fat, high-calorie meal. For BE studies in patients, a different fat and calorie content meal can be used as appropriate for the patient population. Again, this is in line with FDA's current practice. For non-high-risk products, when a fed study is needed, for example, when the labeling indicates to be taken under fed conditions due to the PK reasons, either a high-fat, high-calorie meal or a low-fat, low-calorie meal may be administered, or even a meal which is specified in the labeling could be used. Now, M13A recommends the applicants to provide their rationale for selection of the bioequivalence studies, either the fasting or fed or both, and also the meal type, based on their understanding of the comparator and the test product, whether it's a high-risk or a non-high-risk product. And this is to trigger the applicants to have a better understanding of their own products. Another difference from the FDA's current practice is that the same recommendations with regard to fasting and fed will apply when the studies are deemed necessary to bridge formulations and manufacturing changes during pre as well as the post-marketing phases. And this is different from FDA's current practice, at least for the post-marketing phase. For a level 3 change, needing in vivo study, FDA has generally recommended only one in vivo study. Now the next one, under section 2.2.5, which is for multiple comparators and multiple test products, M13A expands the study design and the statistical analysis of the BioBE studies to support testing of either multiple comparator products from different regions or multiple test products with different formulations in the same trial. So in context of M13A, a comparator product is the product accepted by the regulatory agency for use in conducting a bioequivalence study. For example, for FDA, we have our reference standard, which the applicants would use as the comparator. And expanding the recommendations on the study design to allow multiple comparators to be compared in a single VE study is intended to streamline the bioequivalence demonstration and reduce the cost of the drug development. Now, these studies, they can be designed as a single higher order crossover bioequivalence study comparing multiple comparators against the same test product. With regards to the alpha adjustment, since bioequivalence decisions will be made independently for only the specific comparator which is acceptable within a single jurisdiction, multiplicity correction or alpha adjustment is not needed for the statistical analysis. Preferably, the statistical analysis to be conducted would be 2 at a time. That is a pairwise comparison between the test and the specific comparator of interest for that specific jurisdiction. And however, the study results of all the comparisons performed within the same trial should be included in the clinical study report. The guideline includes similar recommendations to allow for testing multiple formulations of the test product against the same comparator using the higher order crossover study design. And whether alpha adjustment would be needed in this case depends on the underlying objective of the trial. If the objective is to achieve bioequivalence for all test formulations versus the comparator product, then no alpha adjustment would be needed. Here we are not making multiple decisions. However, if the objective is to show bioequivalence of any test formulations, then multiplicity adjustment may be needed depending on the situation. The objective of the trial and the method of multiplicity correction should be pre-specified in the study protocol. The next major change is a new addition. Section 3.4 introduces a new section on pH dependency, where for drug substances with pH-dependent solubility characteristics, an additional bioequivalence study is recommended in certain cases. And although the FDA's PK endpoint guidance does not include this specific recommendation, in practice, FDA has been recommending an additional bioequivalence studies in the PSGs of certain drug products like the palbociclib tablets. So for drug substances showing pH-dependent solubility, the absorption may be influenced by the gastric pH. And bioequivalence under normal fasting conditions between two products may not ensure bioequivalence in a gastric altered pH situation. The test and reference formulations, they may differ in pH stabilizing excipients, the salt forms, or the manufacturing processes between the formulations. And this may alter the gastric pH differently, and therefore it may impact the drug solubility differently. So an additional bioequivalence study with concomitant treatment of a pH modifying drug product, for example, a PPI, would generally be necessary for drugs showing a pH-dependent solubility and dissolution. Now, currently, our knowledge and experience in this topic area is not sufficient. Therefore, details of the bioequivalence study design and the statistical analysis are not specified in this section of the M13A. However, when the guideline is finalized, which would be the step 4 of the guideline development process, The expert working group may likely develop a Q&A document where we would include examples of the study design, the names of the brand drugs formulated with pH modifying or pH stabilizing excipients, comparative dissolution conditions, PBPK modeling, etc. And then the guideline also provides for flexibility. Applicants can justify not conducting an in vivo bioequivalence study by assessing the BE bioequivalence risk based on the totality of the evidence. And the guideline also includes some forms of acceptable justifications which are specified. Moving on, Section 2.2.1.1 introduces removal of the data, and it clarifies the exceptional case where removal of the data may be accepted. So data should only be removed from the statistical analysis based on protocol violations which are contemporaneously documented. And this is the same as in the FDA guidance. When data are removed due to low exposure, in general, removal of no more than one subject study data in each study can be considered. And low exposure is clarified as subjects with very low concentrations, such that the AUC of the subject in one period is less than 5% of the geometric mean AUC of the product in question. Very low concentrations may result from subjects non-compliance, and it may bring the reliability of the dose administration into question. And when subject data are removed, however, all subject data should be submitted, and potential extreme values should be flagged with appropriate documentation as part of the application. This is the last major change that I will cover. Section 2.2.2.3 introduces exceptional cases where potency correction may be accepted with proper justification. And although not stated again in the FDA guidance, exceptions for potency differences marginally beyond the 5% have been considered during assessment if adequately justified. M13A recommends the measured potency of the comparator batch to be within 5% of the test batch used in the pivotal study. And this is similar to FDA's current practice. The potency correction may be acceptable in exceptional cases if its use is pre-specified in the study protocol and it is supported by adequate justification. And the guideline mentions some examples for justification, like including the potency of multiple lots of comparator products, pending market availability, and considering the totality of the evidence. If the justification is found acceptable, then the bioequivalence will be based on potency-corrected data. However, data from both the corrected and uncorrected should be provided. I'll now turn it over to Dr. Kim for the remainder of the presentation. Thank you for your attention.
Thank you, Nilufer. Now we will shift the gear to the other topics that are addressed in the ICH M13A. These additional topics include study population, parent and metabolite, early exposure, dose or strengths to be studied, and other immediate release dosage forms including ODTs, chewable tablets, and oral suspension. With respect to study population, this slide shows the side-by-side comparison of what is recommended in the current FDA BE guidance versus the draft M13A document. Both FDA and M13A recommend healthy subjects in BE studies to reduce variability not related to differences between products. However, the FDA guidance also provides an option of enrolling general population, which is a broad collection of adults with or without stable chronic medical conditions who may or may not be treated with therapeutic drugs that will not interfere with the test medication or bioassay. In terms of intended use and study population, the FDA guidance specifies that if a drug product is intended for use in both sexes, similar proportions of males and females should be included in the study, or provide a justification supporting the use of a single-sex population. However, the M13A simply recommends both male and female subjects to be included in BE studies. With respect to genotype and/or phenotype, the M13A recommends that this information can be considered for safety or PK reasons. While the FDA BE guidance is silent on this topic, FDA's product-specific guidances have incorporated the use of phenotype-genotype information for safety or study design where applicable. For geriatric, the FDA guidance mentions that if the drug product is predominantly intended for use in elderly, applicants should include as many elderly subjects as possible or provide a justification if no elderly subject is included. For pediatric, if the drug product is predominantly intended for use in pediatric patients younger than 6 years of age, the applicant should justify that the BE study results obtained from adult subjects are relevant to the pediatric population, including how the inactive ingredients in the proposed products are appropriate for use in pediatric populations. The M13A is silent on geriatric and pediatric, but mentions of a preference of a body mass index limit of 30. For parent and metabolite, the FDA guidance recommends that only the parent drug is measured because the concentration time profile of the parent drug is more sensitive to changes in formulation performance. The M13A also addresses this. With respect to metabolites, the FDA guidance states that the primary metabolites formed directly from the parent compound should be measured if they are formed substantially through pre-systemic metabolism and contribute significantly to the safety and/or efficacy of the product. The parent drug data should be subject to a confidence interval approach, where the metabolite data can be used as a supportive evidence of a comfortable therapeutic outcome. The M13A states that the demonstration of BE based on the parent drug alone may not be sufficient, and the primary active metabolite should also be considered in some rare cases. Similar to the FDA guidance, the M13A mentions that drugs that have metabolites formed through gut wall or gut lumen metabolism that contributes to efficacy or safety should be measured. This is intended to address situations in which the formulations of the metabolite could be influenced by formulation differences, which may not be detectable when measuring the parent drug. With respect to low concentrations, both FDA guidance and M13A state that BE demonstration can be based on a primary metabolite if the parent drug concentrations are too low to allow reliable analytical measurement. In terms of sampling and early exposure, the FDA guidance mentions of Tmax and partial AUC. The FDA guidance states that the rate of absorption can be assessed by measuring the Cmax, and how the Tmax can also provide important information regarding the rate of absorption. With respect to Tmax, the guidance recommends that Tmax differences should be assessed for any clinical implications. The FDA guidance also states that a partial AUC can be used as exposure measure if specified in the applicable product-specific guidances. The beginning and ending times for the partial AUC should relate to a clinically relevant measure. Similar to FDA guidance, the M13A states that when the early exposure or early onset of action is clinically relevant, an additional PK parameter such as partial AUC, may be used to establish bioequivalence. This partial AUC is typically evaluated from the time of drug administration until a predetermined time point that is related to a clinically relevant pharmacodynamic measure. In terms of dose or strength to be studied, both FDA and M13A recommend the highest strength of the drug product if feasible. The FDA guidance states that use of a lower strength for reasons of safety is generally acceptable if linear elimination has been documented over the therapeutic dose range. The M13A also allows the use of lower strength if the highest strength cannot be administered to healthy subjects for safety and or tolerability reasons, and dose proportionality PK has been documented over the range of strengths. In case where there is a non-proportional increase in AUC and or Cmax with increased dose, the M13A addresses each specific cases given that there may be a difference between different strengths in the sensitivity to detect potential differences between formulations. For drugs with a more than proportional increase in AUC and or Cmax with increased dose over therapeutic dose range, in general, the study should be conducted at the highest strength. For drugs with a less than proportional increase in AUC and or Cmax, BE should be established at the lowest strength if the situation is due to saturation of absorption. If less than dose proportional increase in AUC and or Cmax is due to limited drug solubility, or the reason for non-dose proportionality is unknown, both the lowest and highest strength should be studied. Lastly, the M13A covers other immediate release dosage forms such as ODTs and chewable tablets. For ODTs, the FDA guidance recommends that BE studies be conducted without water if the labeling allows with or without water. However, the M13A recommends that study subjects can wet their mouths with a small amount of water and not allow any fluid intake earlier than 1 hour after administration. Bioequivalence of test and reference ODT drug products taken with water is inferred. For chewable tablet, the FDA guidance states that applicants should administer chewable tablets according to the direction in the reference product labeling. If the labeling states that the tablet must be chewed before swallowing, the product should be chewed in BE studies. If the labeling gives the option of either chewing the product or swallowing as a whole, product should be swallowed whole with a 240 ml of water. In contrast to this, the M13A does not mention of an option of swallowing as a whole, as a chewable tablet should be chewed only. In terms of water intake, the M13A recommends a similar instruction as the ODTs. For oral suspension, the FDA guidance generally recommends that applicants establish BE for a suspension in the same manner as for other solid oral dosage forms. Likewise, the M13 recommends that bioequivalence studies be conducted according to the reference product labeling. For other oral formulations, the FDA guidance states that the sublingual tablet should not be swallowed, but placed under the tongue until dissolved. The M13A recommends that a similar approach is taken as for ODTs. As for the implementation, this ICH M13A document is a draft form, and it is currently under public consultation. Therefore, it is not ready for adoption or implementation. With respect to the current M13A draft, we encourage stakeholders to submit comments to the regulatory agencies so that the FDA and ICH M13 Expert Working Group can consider your comments before they begin to work on the final version of the guideline. In the interim, applicants should refer to the currently posted FDA General BE guidance and product-specific guidances. As one is aware, FDA guidance do not establish legally enforceable responsibilities. Applicants can use an alternative approach if it satisfies the requirement of the applicable statutes and regulations. If applicants propose an alternative method different from the guidance, they can provide justification, submit control correspondence, or meet with FDA through proper pre-ANDA or ANDA meetings. In our presentation, we provided an overview of the harmonized ICH M13A guideline, which provides recommendations on BE study design, principles for conducting BE studies, and BE standards for immediate release solid oral dosage forms. This harmonized M13A guideline should reduce the need for additional in vivo BE studies, such as a fat BE study, reduce need for conducting multiple BE studies for multiple jurisdictions, and support streamlined global generic drug development. The M13 Expert Working Group will review submitted comments during the public consultation period before finalizing the M13A guideline. The efforts of harmonizing the Tier 2, which covers a bioequivalence for additional strengths of a product line including biowaiver considerations, and the Tier 3, which covers highly variable drugs, drugs with narrow therapeutic index, and complex BE study design analysis for considerations, will continue with M13B and M13C. Now, moving on to challenge questions. Please take a moment to consider the following two questions. Question 1. What is the most significant difference between recommendations in ICH M13A as compared to FDA's current draft ANDA BE guidance? A. Recommendations for fasting versus fat BE study. B. Bioequivalence for long half-life drugs. C. Multiple competitor products. Or D. Batch size for pivotal BE studies. The answer is A. Recommendations for fasting versus fat BE study. Question 2. Which of the following products are not covered in M13A? A. Immediate release oral products. B. Modified release oral products. C. Orally disintegrating tablets. D. chewable tablets. Or E. granules, powders for oral suspension. Of course, the answer is B. Modified release oral products. Lastly, here are the additional resources for your reference. With that, this concludes our presentation on navigating the first ICH generic drug draft guideline, M13A bioequivalence for immediate release solid oral dosage forms. Thank you for your attention.
Thank you for those great presentations. We'll now move into our panel discussion. Moderating the panel discussion is Dr. Sarah Ibrahim, Associate Director for Stakeholder and Global Engagement in the Office of Generic Drugs. Joining us on the panel discussion, we welcome David Coppersmith, Regulatory Counsel, Office of Generic Drug Policy and OGD, Dr. Stella Grosser, Office of Biostatistics, Office of Translational Sciences. We have a returning presenter, Dr. M.J. Kim, and we also welcome Dr. Robert Lionberger, Director of Office of Research and Standards in OGD. Also, Dr. Parther Roy, Director of Office of Bioequivalence in OGD. And finally, Dr. Ethan Steer, Associate Director of Lifecycle Management, Office of Clinical Pharmacology and OTS. Welcome back our final two presenters, Dr. Nilufer Tampal and Dr. Lei Zhang. Please join me in welcoming our panel moderator, Dr. Ibrahim.
Thank you for the kind introduction, and thank you for the wonderful speakers on their presentations today. Let's go ahead with the first question for our discussion. So what is FDA's plan in implementing M13A? And we can really think of that question in two sections. And currently, if there is a difference in recommendation between draft M13A and FDA's PSG, or general guidance, should applicants follow FDA's PSG, general guidance, or M13A? And of course, what is the plan for future after M13A is finalized? And I would like to invite Dr. Roy to start that question.
And Thanks, Sarah. Yeah, so I think this is probably a great question, and I know it's probably in everybody's mind, right? Just to provide some introductory comments on M13. M13A is not an established guideline yet, right? It is out there for public consultation. So if you like the concepts that are being discussed within M13A, hopefully you have provided your comments for on M13A that you kind of like some of the concepts that are published. So in going back to FDA's PSG and general guidance, I think those are the guidances. Those are the established guidances for the agency, and that's what I think you should follow. Having said that, I mean, there are obviously some scientific principles laid out in M13A, which may be slightly different from the general guidances, as the speakers have nicely talked about. Then obviously you are welcome to make some of the scientific justification within the application, either come as a topic for a meeting discussion or in the control correspondence. So those are you can certainly make some of those scientific justification laid out to the agency, but you will not. But again, I mean, just to remind everybody that M13A is not a guideline right now, and it has not been implemented yet. We are going through a consultation process, so you will not be able to sort of say, you cite M13, oh, based on M13A, here is my proposal. I think that's probably not going to work, but certainly you are encouraged to utilize some of the scientific principles that are laid out in this new ICH guideline. So I will stop there. Thank you.
Thank you very much. I'd like to ask Dr. Kim to maybe follow up on the question.
Sure. Thank you so much. And hello, everyone. First of all, I want to share my comments regarding today's webinar. I am so excited to see so many of you participating in this webinar, and it shows the great amount of interest and excitement about this ICH M13A document. So with that, and I have to echo what Partha just mentioned. With respect to the M13A document, as Partha mentioned, it's not a finalized version. And secondly, this is not the FDA's document. This is international document. So that means that all your comments will be extremely important for the ICH M13 expert working group members to see and review before they can be finalized. So as you heard from Dr. Lei Zhang's presentation, in the earlier slide, there were still a number of regulatory agencies there who are still accepting comments from you. So we strongly encourage you to submit your comments and whether they're positive or negative. And if you have some alternate approach, I welcome you to submit those comments as well. And what we are really excited to see is that a good number of you already have submitted lots of comments to the docket, as well as for our webinar. I see that there are many questions and comments from your end. So I really hope that you submit those comments to the regulatory agencies that are still open for your comments to be considered. So with that said, my division, we actually, we are primarily responsible for working on the product-specific guidances for the immediate release drug products. So what we intend to do is that once the M13A document is finalized, we are going to make sure that our PSGs published by FDA will be in alignment with what's recommended in M13A document. And until this M13 document is finalized, you need to follow our current FDA BE guidance, as well as what's in the product-specific guidance. So with that, I will stop here. Thank you.
Thank you very much. We can move on to the next question. So although the draft M13A proposes both fasting and fed bioequivalence studies only for high-risk products, currently FDA recommends both fasting and fed BE studies for almost all oral immediate release products. Now, for an application that will be submitted before M13A implementation, can one of the BE studies be waived? And if so, what data may be used to support the justification? And I'd like to invite Dr. Leinberger to comment on the question.
So thank you, Sarah. So happy to participate in this fantastic webinar. I think this question is very similar to the previous question about implementation. And again, if you are interested and you think the approaches that are in the M13A public comment version are valuable to you and your development program, please make that public comment to the agencies so that the things that you want to do or might want to do and you might want to include in an application actually do get into the final version of M13 that's implemented. So again, like we really want to say that that's the appropriate part of the process here. This is really a version that's out for comment. So it's really not useful for you to submit an alternative approach justification to FDA citing this draft M13A. If you want to make mention of that, please provide your comments to that. Once it's implemented, as MJ mentioned, we'll be updating guidances on the product specific level to make it clear, what FDA thinks are high risk products for the fed versus fasted bioequivalence studies. So again, certainly, at any time you can, if you read any of our guidances, it says our guidances are recommendations, and you can submit alternative approaches to them with scientific justifications. But again, citing the draft M13 is not going to be it's not a well-received justification. Really, we want you to go through, and as Lei described in the introduction, for the ICH process, there's a very well-defined comment period. Once that comment period is over and the working group digests the comments, then the versions come back for adoption by each individual regulatory agency. And that's the point at which this becomes FDA's recommendations for studies. And in that case, that's something that you can say, well, look, FDA's recommendation says, this study is not needed. And I think that, both of the first questions for the discussion address this issue because, we do think this is a major shift in the reduction of the burden of BE studies for products. But it's something that really, because of the scale and the impact that it can have, it can only be really implemented efficiently through a planned process of implementation. And if applicants come in and say, well, can I implement M13 now because it's just a draft and that's my reason, that's really not appropriate. And it's really not the way to run an efficient program here for this scale, this type of scale and change of this impact. So again, we through 5 or 6 years ago at our public workshop on science and research priorities, we heard very clearly, we solicited a lot of scientific comments on the need for fed and fasted BE studies and under what scientific considerations were made. We asked, many generic companies provided data from their development programs on this question that fed into our understanding and fed into the ICH process. So again, this is a significant change. It's a very impactful change. I think it's very valuable to the generic industry, but we strongly encourage you to follow and support the ICH process for doing this change and making this change broadly applicable and not wasting effort on sort of one-time requests for specific applications.
Thank you for that. And Dr. Tampal, can you please comment on question 2?
Yeah, thank you, Sarah. Yeah, sure I can. I would also like to start by reiterating what my other panelists have already said here. We are still far away from implementing the guideline, and there is a process that the ICH guideline development has to go through. But coming to the specific question of whether a study can be waived, it will depend on the scientific merits of the justification that the applicant may provide to us. Because applicants will need here not only to have a good understanding of their test product, but also the comparator, how that is formulated, what are the critical aspects of those formulations. FDA, we always encourage and we are open to supporting applicants' proposals for alternative approaches if these are backed by scientific rationale that FDA finds acceptable. And that will hold true even in case where the applicant is wanting to consider to adopt the scientific thinking from the ICH guideline and applying it to the development program. So then based on your understanding of the formulation characteristics of your own product as well as the comparator, the differences in the excipients and how they will impact the solubility or the permeability or the dissolution, including also the biopharmaceutical properties of the drug substance, you taking everything into consideration, you may be able to justify that a formulation-dependent impact on the drug absorption would not be expected for your product under the different GI conditions of the fasting and fed state. So that option is always there for you. And then going forward, once M13A is adopted, based on the available information of the comparator product, our PSGs will indicate whether only one study will suffice or both fasting and fed would be needed. So I hope this helps to answer this question and better your understanding. Thank you.
Thank you very much. Moving on to question number 3. So when only a bioequivalence study conducted under fed conditions is needed for a non-high-risk product, why are there multiple options for meal types that are recommended in M13A, and that a low-fat, low-calorie meal is acceptable in addition to the high-fat, high-calorie meal traditionally employed for studies conducted under fed conditions? And I'd like to invite Dr. Zhang to respond to the question. I think we have a technical. Thank you for that. Dr. Zhang, are you able to reconnect to the meeting? Okay. Or Dr. Nilufer Tampal, would you like to comment on question number 3?
Sorry, I just back. Is this better?
Yes, thank you.
Thank you. Sorry, I think I have some issue with the headset. So I will just restart. So sounds like I think this question is really to address there's a new element in M13A which for the non-high-risk product when you only need to conduct one study, which is a Fat-B study, and we give multiple options, including a low-fat, low-calorie meal to be acceptable in addition to high-fat, high-calorie meal that is traditionally used. So I think this will be a good opportunity to explain the thinking behind and discussion occurred at the expert working group. So we all know traditionally the high-fat, high-calorie meal is being recommended for food effect or Fat-B studies. The reason we know that is that those classic high-fat, high-calorie meal is expected to provide the greatest perturbation in the GI tract as compared to the fasted condition, so-called maybe worst-case scenario, which along with the fasted condition would cover the whole spectrum of the GI conditions to understand the food effect or what food may affect the BE. So M13A, also you heard that M13A do recommend both fasting Fat-B studies for high-risk product for which the food can, we expect to have a differential formulation-dependent impact on the absorption of the drug substance from those high-risk products. And if the Fat-B study should be for high-risk products should be conducted with a high-fat, high-calorie meal. So this will ensure we have B data at both ends of the spectrum of the GI tract conditions. And also by utilizing this approach, our expectation is that the BE outcomes from these two conditions would cover the BE under GI tract conditions probably created by any meal types, including low-fat, low-calorie meal that could fall somewhere between the fasting and the high-fat meal conditions. However, for the non-high-risk product, this logic does not hold because for non-high-risk product, we do not think the food conditions will affect the BE outcomes based on the understanding of the formulation and how the food may interact with the formulation. Therefore, for non-high-risk IR products, we generally acknowledge that a 1B study is sufficient, and typically a fasting B study is preferred condition to study BE because this will, the fasting condition usually provides a greater discrimination or more sensitive between to detect the formulation difference on PK. This is why we recommend 1B study under fasting condition is sufficient for those non-high-risk product in most cases. However, for some non-high-risk product, if we recommend that 1B study should be conducted under fair conditions, for example, due to safety reasons or due to the labeling may recommend to be conducted, to be taken with food due to PK reasons, then M13 expert working group proposed the use of a low-fat, low-calorie meal as one of the options to better approach and the minimal GI perturbation while still addressing the need for food. We also consider this more moderate meal, which covers the with food requirement is less extreme in terms of the impact on the GI tract, and also could be more realistic in terms of the meals that a patient is likely to consume. So the goal of the meal here is not to create the worst case scenario for checking the possible B failure at risk, because we do not expect that different meals will lead to different B outcomes for those non-high risk products. Therefore, a low fat, low calorie meal is acceptable here. And the reason we also offer other options is mainly to offer some flexibility in the B study design, consider the product to be studied. And also some labeling may have very specified the meal types in the labeling, which we think that meal type should also be administered, maybe, which makes sense. So again, this is a new element, I believe. And M13 EWG think this is a reasonable proposal moving forward. So we also look forward to the comments that may be received through the public consultation period. Thank you.
Thank you for a very thorough response that is truly scientifically driven. Moving on to the next question. How may M13A, after it is finalized, be applied to new drug applications that may rely on BE for decision making? And Dr. Ethan Steyer from OCP, can you please comment on the question?
Thanks, Sarah. And can you hear me okay?
Yes, we can.
Okay, great, thanks. Thank you. So I just also want to say I'm also excited to be here to join my colleagues from Office of Generic Drugs to talk about M13A today. So I just want to address this question in a couple of points. One, to echo back to Dr. Leinberger's comments and Dr. Roy's comments at the beginning. I just want to emphasize this guidance is in draft form. So, it's not possible to get into the details, the nitty-gritty of how this will be applied on a case-by-case basis today. But again, really echo the comments earlier to encourage everyone to submit their comments and or questions to the docket. So that can be incorporated in the discussions as they move forward, hopefully leading to the finalization. I also want to highlight in the draft guidance that there's a really nice section in lines 52 to 58 of the guidance that outlines the scope of this guidance and basically talks about some scenarios where this will not apply. I'll just repeat that again, will not apply to the new drug side. Those include, among others, relative bioavailability studies, food effect studies, and bridging studies without the necessity to demonstrate BE. So I really want to highlight that to audience members today to look at lines 52 to 58. I also want to highlight too that once this guidance is finalized in whatever final form it takes shape, that on the new drug side, sponsors will be able to discuss with the review divisions, during the IND stage, how, when, if they need to conduct a BE study that would be covered by this guidance as opposed to a comparative bioavailability study, which is typically the more common study conducted in OCP. And I really want to keep the focus on generics today as OGD is hosting this discussion and the large impact this guidance will have on their side. But I just will make a brief reference. There was recently an SBIA on the recently finalized comparative bioavailability guidance, as well as being able to find that guidance on the FDA website. So I just briefly mentioned that to direct audience members interested in BA studies to refer to that. And I'll pass it back to you, Sarah. Thank you.
Thank you. We can go ahead and move to question number 5. So the FDA's current ANDA BE guidance partially overlaps with M13A, with more content that will be harmonized in the future. What is FDA's plan on the revision of the ANDA BE guidance when M13A is finalized and ready for implementation in the near future? And such a wonderful policy question. I'd like to invite Dr. Coppersmith.
Thank you for the question, Sarah. As was discussed during the presentation, many of the topics covered by the current ANDA bioequivalence guidance, the bioequivalent studies with pharmacokinetic endpoints for drugs submitted under an ANDA guidance that was published back in August of 2021, are covered by M13A. And as was discussed during the presentation, there are some major differences between the two. In addition, there are some topics that are covered by the current ANDA bioequivalence guidance that are not discussed in the M13A guideline. This includes, for example, modified release products, which will be discussed in the upcoming M13B guideline. And there are other topics covered by the current ANDA bioequivalence guidance, which are not planned to be discussed in either the M13B or M13C guidelines. And it's a slightly different situation with M13A than with the ICH M9 guideline on BCS-based biowaivers that was published back in 2021, where the topics covered were the same as the previous FDA guidance on that topic. So once M13A is finalized and adopted by FDA, it will reflect FDA's current thinking on this topic, and FDA will take the actions that the agency deems necessary to align the recommendations in the current ANDA bioequivalence guidance with the recommendations included in the final versions of M13A to ensure that the recommendations are consistent and that there's not unnecessary overlap between the guidances. And also in the future, as other FDA guidances are updated, references will be updated to the appropriate final M13 guidances where applicable. So thank you.
Thank you for a thorough response. We can move to the next question. So under multiple test product scenario, the guidance states that if the objective is to show BE for any of the test formulations, then multiplicity adjustment may be needed. Can Dr. Stella Grosser please comment or give an example on when it is needed and when it is not needed?
Sure. Thank you so much for this question. And again, I'm happy to be here like everyone else. First, I want to reiterate what Nilufer said in her description of these sections. The essence of this question, of this problem, is the decisions that are going to be made. It has less to do with the relation between formulations or products and more about the objectives of the trial. So an example of when multiplicity adjustment likely would be needed is if you have two test products, two different formulations, and you're not sure which one you plan to market. So you have a three-way crossover. You're testing the reference, the comparator product, against each of these formulations. And if either of them are shown to be E, then you will decide after the trial finishes which one you will submit for for marketing approval. On the other hand, if you have two formulations and you test it in a three-way crossover against the single comparator, and you will only submit when, if and only if both are shown to be, both test formulations are shown to be bioequivalent, then you will not need to adjust for multiple comparisons. It has to do with the, again, nature of the decisions. And that's why for multiple comparator products, the situation is different because, any one application would be based on that region's comparator product and the proposed test generic product. So let me end with that, and we can. Oh, and actually, I do want to say that this is a harder question than it may appear. And so we welcome comments. Please submit them, comments, questions, and also scenarios that you might be thinking of, like, well, what if it's this, that, and the other thing? Specify this, that, and the other thing, and what the objectives would be and what the situation would be, and then we can think this through some more. Thank you very much.
Thank you, and you raise an excellent point. Thank you. You raise an excellent point. The ability to comment and propose these scenarios is helpful. And yes, the question does seem a lot simpler than it really is. I can see from all the attendees and the questions in the Q&A section the significant interest, and it is understood, of course, that this is the first generic drug sole ICH guideline, and it does provide harmonized scientific recommendations for conducting BE studies during both development and post-approval phases. We understand that with such guidance, we can increase the efficiency of drug development and accelerate availability of safe and effective orally administered IR solid oral dosage forms. With that, I'd like to switch it over to Ray Ford for the Q&A session.
Thank you so much for the panel discussion and the presentations. We'll now transition to our Q&A session. As a reminder to our attendees, if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. Looks like we do have some questions coming in. The first group of questions will be addressed to Dr. Lei Zhang, and the first question will be a combination of 3 questions that we're going to try to put together into one question. And here's the question. What is the procedure to join the ICH Expert Working Group? And has TGA not published the M13A for public comment? And also another question in regards to joining and commenting is, why has the African regulatory authorities not involved in the public consultation?
Okay, thank you for these serious questions. They are all kind of ICH process related. So thank you for those questions. I'm not an expert on ICH process. I can just comment on based on my experience working on the expert working group so far. So I think in terms of the process for joining the ICH, I would refer the people to the ICH website, which is www.ich.org. They have document to explain their process and the procedures. And you can also see the list of current ICH members and also observers. And also the group that will be joining the expert working group will be from the ICH, either members or observers. So in terms of the ICH M13, we have 12 regulatory members and some of the industry groups and also observers, members and observers. So only the members regulatory agency will publish the guidance for public consultation. That's why you see the list of those 10 with their respective dates. And TGA and also we have Jordan FDA in the working group. They are observers, they are not a member. And that's the reason why they are not like published for public consultation, but they do have their own process to solicit comments. So I would defer the people to reach out to their regional, either the agency or members for if they want to submit the comments to find out how to get the comments across. So I will just end here. Thank you. Oh, and another thing is, I know recently SBI does have a webinar that is held jointly between Health Canada and FDA on February 24th this year. They also have a session on the like an introduction of the ICH. So people may also refer to that presentation for more information. Thank you.
Thank you, Dr. Zhang, for responding to that question. We do have a few more, and here's the next question. Some products have been classified as BCS1 by the EMEA guidance, but not by FDA. Do you think there is going to be a harmonization among all agencies? Dr. Zane is the moderator. If you're responding, your mute is on, so you please check your mute.
Thank you, Ray. So this question is not really M13 related. So BCS classification and also BCS base waiver is covered by another ICH guidance, which is M9. It has already been adopted by multiple regulatory agencies, reached step 5 in 2021 by the FDA. So I think because this guidance is in place, I don't know what specific product this person is referred to. I think by following the M9, there should be a high likelihood of the harmonization on the product, how they are being classified as BCS1. Thank you.
Thank you for responding to that question. We have one more question that just came in for Dr. Zhang, and here's the question. Should pilot PK studies be listed in Module 2.7.1 and the complete study report be submitted in Module 5?
Thank you for the question. I think we do have some explanation in section 4 of the M13A regarding the documentation. So the PK studies, pilot PK studies should be listed in module 2.7.1. However, the complete report is not needed in module 5 because they are pilot study, typically probably not rely on to be for the decision making. However, the full report should be available if there's a request from the regulatory agencies. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist. Got a few questions that came in for Dr. Nilufer Tampal, and here is the first question. In determining high-risk products, do you expect that the labeling of the comparator product across various countries would be consistent? And if not, how would we align across various versions of the labeling?
Thank you, Ray. Yeah, that's a good question. But unfortunately, for the ICH guideline, the M13A, labeling harmonization is not within the scope. So in most cases for the IR products, we would expect similar labeling. But if it is not, that's not something that we would be working to even harmonize. So in that case, I think we would follow the recommendations in the labeling for that particular jurisdiction. Thank you.
Thank you for responding to that question. Our next question for Dr. Tampal is the following. For an amorphous solid dispersion product, which is being categorized as a high-risk product, is a fasting and fed study sufficient, or should also a PPI study be required if the brand label indicates no impact of PPI on bioavailability?
Yeah, that's another good question. I would say the innovator, so if it's in the labeling of the comparator or the innovator's product, they would have taken some specific steps to avoid the effect of the drug on that absorption. So under different GI pH, they would have taken some measures by probably using pH stabilizing excipients or different salt forms or whatever. But if the test now has different excipients or they are using a different manufacturing technology, the features of the innovator's product may not be reproduced by the test product. Because we know for the same API, different formulation techniques can have a different dissolution characteristic at the elevated pH, which is what we expect when in presence of the PPI. So no, the answer is we would need to assess the risk of bioinequivalence for these products at elevated pH. Thank you.
Thank you for responding to that question. A couple more questions came in for Dr. Tampal, and here's the next question. Until the time that the ICH M13 is formally adopted, will there be any flexibility in not conducting fed studies for products that are low risk if the justification for the same is provided by the applicant?
Yes, thank you. I this question, I think I already, in a way, answered it for this panel question. Like, yes, if you, it will depend on the scientific merits of the justification that you provide and whether that's acceptable to the FDA. And as I said, those options are always still available to the applicants. So yes, the answer, I think I've already answered this question. So maybe we can.
Thank you for responding to that question. We have one more question for Dr. Tampal in this round, and here's the question. In a three-way study with two different countries' RLD versus the single test product that failed against one of the RLDs that was non-US, would that impact a decision on and approval?
No, so that would not impact, I would say, in the decision on the ENDA because the test product against the foreign RLD, that's something which will be of our interest from a scientific standpoint, but it's not something that we would be interested from an approval standpoint. So that would not be any kind of an impact for ENDA approval. Thank you.
Thank you for responding to that question. Moving on to our next presenter, we've got a couple of questions that came in for Dr. MJ Kim. And here is a combination question. We're going to try to put two questions together. And here is the first part of the question. Based on the M13A, once implemented, the guidance, are equal proportion of males and females not necessary? And is there no need to justify either in the end application? And also, if there's no known gender effect on a product, then is it necessary to consider both the gender population during the conduct of the BE study of this product? And if yes, what ratio should be considered?
Yes, this topic has been on our mind for a while since the FDA's and the BE guidance was published. And to our surprise, in a way, we have received a good number of controlled correspondence on this topic. So what I want to kind of use this time to clarify and comment on this topic is that one needs to understand that when a drug product is intended for use in both sexes, then that means that women should also be included for the assessment of bioequivalency. However, we are also aware that there may be some concerns due to the safety of females of reproductive potential, or maybe there is some cultural issues in terms of including females. But our overall goal is that the applicants try their best to recruit female subjects in the BE studies if a drug is intended for both sexes. So with that said, the M13 draft guideline does not have that language about similar proportion of males and females, but that guideline still recommends that both females and males are included in BE studies. So I want to kind of reiterate the importance of including females in these studies if the drug product is marketed to be marketed in both sexes. And in terms of the ratio, or if there is no so-called gender effect, and those of you who know me, we get a ding for using the term gender because it should be sex. One cannot change the sex. So we are actually assessing the sex differences, not the gender difference. But if there is no so-called sex differences with the drug product, but one can refer to the RLD product labeling to see whether there are any kinds of PK differences or any safety differences in these two populations. But overall, regardless, our recommendation is that you try to enroll many female subjects as possible. And that doesn't, so that kind of precludes in terms of putting that specific ratio and all that. And I guess the FDA's current guidance, when we mentioned about equal or similar proportion of females and males, that kind of generated lots of discussions or questions from the applicants. But overall, our key point is try to enroll many female subjects as possible in these studies. Thank you.
Thank you for responding to that question. We've got another couple more questions for Dr. Kim, and here's the next question, and it's regarding slide 48. So regarding slide 48, where the Tmax and PAUC are mentioned, does the difference indicate a de-emphasis on Tmax or more emphasis on PAUC?
Sure. So one needs to be mindful that the FDA's and the BE guidance does not necessarily cover just the immediate release drug products, but it covers the other products such as modified release drug products. And that FDA guidance talks about the Tmax in number of sections. So I specifically put that information. But as one can imagine, with the immediate release drug products, because the PK profile is relatively quick compared with modified release drugs, it's kind of questionable how to utilize the value or the utility of Tmax, because there will be potentially highly variable type of Tmax, and whether there are really a need for the Tmax assessment. But ultimately, the other key question is, even if we think that Tmax assessment is extremely important, then the question is like, what statistical methods should we use to do comparison of the Tmax of reference and the test products. So those were like some of the talking points that we actually discussed extensively within the external work group. So I'm not necessarily saying that the Tmax is de-emphasized, but just pointing out that the FDA guidance, where we talk about Tmax, it applies to other dosage forms. But when it comes to M13, the document, we kind of give example as a partial AUC for the early exposure comparison. So we're not necessarily saying that the partial AUC is the only matrix that can assess the importance of early exposure when there is clinical implications, but partial AUC is one of. And as of now, I cannot think of any specific IR drug product where we have a partial AUC for BE assessment at this point yet, but there will be some products that we may consider looking into it. But at this point, I would say some drug products with early onset or pain management type, maybe some of that, some of those products that fit into the category. But once again, I'm not trying to de-emphasize the Tmax, but there are some challenges that associated with Tmax and statistical comparison. And for the early exposure, the use of partial AUC may be one of the matrix to use. Thank you.
Thank you for responding to that question. We have got a couple of questions that came in for Dave Coppersmith, and here is the first question. Can a comparator product be another generic drug on the market instead of the branded product?
So thank you for the question. For FDA, the comparator product that an applicant can use to compare against the test product in conducting an in vivo BE study is the reference standard. And ordinarily, FDA selects the reference standard or the reference listed drug as the reference standard. Where FDA cannot select the reference listed drug as the reference standard, for example, where the RLD has been withdrawn from sale for reasons other than safety and effectiveness, FDA generally will select a previously approved ANDA that referred to and is therapeutically equivalent to the reference listed drug as the reference standard. And the reference listed drug and reference standard designations are included in FDA's approved drug products with therapeutic equivalence evaluations publication, commonly known as the Orange Book. And so you can refer to that publication to find the RLD and the reference standard.
Thank you for responding to that question. We've got a couple more questions for Dave Coppersmith, and here's the next question. Are applicants required to follow the ICH guidelines such as M13A?
So as my colleagues have mentioned, the draft M13A guideline was issued for public consultation for comment. When M13A is finalized and adopted by FDA, it will reflect FDA's current thinking on this topic. It will contain recommendations, not requirements, and applicants can use alternative approaches as long as those approaches comply with the applicable statutes and FDA regulations. Thank you.
Thank you for responding to that question. We have one more question for Dave Coppersmith, and here's the question. Can I still submit comments on M13 to FDA with the comment period having closed, or do I have to submit comments to one of the other ICH member agencies whose comment period is still open?
So thank you for the question. That's a great question. So as was discussed during the presentation, the comment period for FDA closed in early April. However, per FDA's regulations, and as stated in the notice of availability for the M13A guideline that was published in the Federal Register, interested parties can submit comments on any guidance at any time to FDA. However, to ensure that comments are received and considered before work begins on a final guidance, comments have to be received before the comment period closes. Therefore, to ensure that your comments are received and considered before ICH begins working on the final version of M13A, if you've not already submitted your comments, I would recommend that you submit your comments through one of the ICH member agencies whose comment period is still open, and that information was included on one of the slides in the presentation, or through the ICH Secretariat. And you can refer to the ICH M13A website for more information on submitting comments through the ICH Secretariat, including the relevant email address, which is step2 and then comments @ich.org. But again, that website's listed on the ICH M13A website. And comments submitted through that email address are then forwarded to the ICH expert working group. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we've got a few questions that came in for Dr. Stella Grosser. And here is the first question regarding BE studies. What group should be a variable in the statistical model if it's not expected to have an impact on the response?
Hi, thank you. Group often has impact on the response, and depending on how your group is defined in your particular study, and so we expect that to be checked. So it may well have an impact. Yeah, thank you.
Thank you for responding to that question. We've got another question for Dr. Grosser regarding a mixed effect model. And here's the question. In a mixed effect model, that it's not specified if in the draft M13A, only an ANOVA is mentioned. Isn't it appropriate to include?
Yeah, thank you. The ANOVA is mentioned as an example, and certainly if it's appropriate to have general model, random effects included for the subjects. By all means, pre-specify and model your data that way. Different regions have different practices, and we're still in the process of harmonizing.
Thank you for responding to that question. We have one more question for Dr. Grosser, and here's the question regarding a parallel design. If randomization is already stratified by a known relevant factor, can you explain why the factor should be included in the model, and what are examples of relevant covariates?
So there's a saying in statistical analysis of clinical trials, which is analyze as you randomize. It's not a mathematical requirement, but the point of putting those stratification factors in the subsequent model for analysis is that you get more precise estimates of your effects, and in particular, the treatment effect will be, and in this case, the treatment effect is, the question of the equivalence between your products. That will be better estimated, more precisely estimated, and so the confidence interval will be smaller and more likely to fit within the equivalence limits. So that's the sort of thinking there. As far as covariates go, I would say for immediate release products, there are, something like BMI might be such a covariate. Site, depending on the product again, maybe the clinical site might be important. And, different sorts of products have different sorts of demographics that might affect the pharmacokinetics. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we've got a couple of questions that came in for Dr. Partha Roy. And here is the first question for Dr. Roy. Will FDA apply ICH M13A changes to ongoing development programs or pending applications after M13 is in implementation?
Yeah, thank you for that question. I think this is a great question, and I think this question is probably in a lot of people's mind. Yeah, once the the implementation happened and it gets finalized, and again, for the pending applications, I'm assuming that you may have submitted information and make a scientific case for maybe the reducing of the certain burden of doing an extra study or something like that, and that is consistent with the M13 scientific concepts that are laid out in the guidance. So we will definitely take a look at it once it is final and on the pending applications, and we'll probably have a plan on how to kind of apply that. That we do for all guidances, for all of the FDA guidances. We do take a look at all of the pending applications and how that impacts. Hopefully, the scientific principles that you laid out in your application, consistent with some of the M13A thinking, is going to be reviewed based on the science and based on the scientific justification you laid out, whether the M13A gets finalized or not. I mean, that's always that's what we do all the time. Again, it is the guidance tells us our current thinking on some of the scientific and the regulatory concepts that are within the guidelines. So yes, the answer is yes. We will take a look at and we'll also look at how that impacts the pending application and ongoing development program. Thank you.
Thank you for responding to that question. Another question came in from Dr. Roy, and here is the question. Is the classification of high-risk products known to industry or only internal to the FDA or other regulatory authorities?
Yeah, again, thank you for the question. I think that's a great question. I don't think we have a list of high risk products as such at this time. And I don't think anybody outside also has a certain list, but I think you heard from the presentations that there are products which may have complex formulations or manufacturing technologies that are being used and which ultimately will define whether that is a high risk. Again, I wanted to redefine high risk for demonstrating bioequivalence. So the question is, we will go back and it's going to be a case by case determination. And if you think that uh, the question really is that whether your test product or the product or your reference product, whether it's a high risk or even low risk for that matter, if you want to define it that way, certainly we can have that discussion again based on the technology that has been used, the formulation and the manufacturing technology that has been utilized within the program. So again, there is not a list that we can go to at this point, but you also heard that once the M13 gets finalized, we will definitely take a look at it and probably go back and update some of the product specific guidances based on some of the known products, based on some of the known reference product. So and we'll go through that exercise once it is final. But at this time, I don't think the agency or any of the agencies or the industry has a certain list that you can just go and take a look at, which is a high risk and a low risk product. Thank you.
Thank you for responding to those questions. Moving on to our next panelist, we have a couple questions that came in from Dr. Ethan Steer. And here is the first question. Will the M13 guideline also apply to 505B2 as much of the info relates to the procedures in conducting a study?
Thanks, Jeff. So first, I just want to refer, the audience back to the, recording when it's released, some of my earlier comments that I won't repeat here. But, I think it's important to keep in mind, there is a subset of 505B2 applications where applicants seek a therapeutic equivalence code, somewhat oversimplifying here, but similar to generic. So, I think that's a case where, there would be a clear discussion about using the M13. But, in terms of, the details and specifics of how it's applied in these contexts, I think it's really important to, again, engage in those discussions. And also, as I mentioned earlier, refer back to the comparative bioavailability guidance, which is typically the more commonly conducted study on the new drug side. So yes, thanks.
Thank you for responding to that question. Moving up to the beginning of our panel, we've got a few more questions that came in for Dr. Lei Zhang. And here is the first question for Dr. Zhang. If the purpose of the M13A is to harmonize and reduce the need for multiple different sets of data and information from duplicative PE studies, then further harmonization on use of foreign comparator reference products should be sought. Is there a reason why ICH did not recommend use of a foreign reference products?
Thank you for the question. I think this is probably in a lot of people's mind. ICH is mainly focused on the scientific and technical experts of the harmonization. Use of foreign comparator or what may be suitable as a comparator for certain B studies is really governed by the regional legal framework. So ICH, it is out, as we mentioned, this is out of the scope. We do recognize the value of reducing the burden for additional study. That's why in the M13A, we do attempt to recommend the multiple comparator studies, which leading towards some more harmonized or reduced study for including multiple comparators. So to build a scientific bridge. So this is just a scientific attempt, but with regard to the recommendation of the foreign reference product, this is really out of the scope for M13. Thank you.
Thank you for responding to that question. Moving on to the next question for Dr. Zhang. And here's the question. For a low-fat, low-calorie meal, how many calories should be considered? And are there any examples of such a meal, and will it be made available in the guideline?
Yes. For the guidance, we did have one like a sentence give an example of what low calorie or low fat, low calorie meal is, which is in section 2.1.5. Have that definition, which is about like a meal of approximately 500 kcal with approximately 25% calorie from fat. I think we probably do not want to include a lot of details in the guidance itself. However, when the guidance is at the step 4 in the finalization, after it's finalized, a Q&A document can be developed, and we can decide this may be a topic to be included in the Q&A, where we will have room to provide more examples and clarification to the stakeholders. Thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Zhang, and here's the next question. For high-risk products, if the labeling mentions to take under fasting, then why is testing under the fed condition needed?
Yeah, this is a very good and interesting question. I think this is probably in a lot of people's mind. As you probably also heard Dr. Nilufer Tampal also comment on, the labeling is currently not really harmonized. So if we need to decide why that particular product, the labeling recommended to be taken under fasting condition, whether it's due to safety reason or some other reason. If it's due to safety reason, probably the fat is not needed, even for the high-risk product. However, if it is safe to be taken under the fat condition for those high-risk product, the reason the guidance currently recommend both fast and fat study is to be able to get the data from both fast and fat conditions, that therefore we can cover the whole spectrum of the BE for those high-risk products, where we know the BE outcome may be different between the test and the comparator. So this is the main reason why we recommend both fast and fat studies for those high-risk product, irrespective of the labeling recommendation. Thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Zhang. Are the M13A, B, and C going to be standalone documents or one document with 3 major sections?
Yes, I think we currently, the plan is to develop those 3 as individual documents rather than different sections under one big document. So it will be a series of guideline, which will be standalone documents covering each of the tiers of the topics. Thank you.
Thank you for responding to that question. We have one more question for Dr. Zhang, and here is regarding the presentation. BE studies for highly variable drugs and drugs with a narrow therapeutic index will not be addressed until M13C, which will not be complete for quite some time. Given that, what will the expectations be for those types of studies in the various regulatory jurisdictions during the time between the adoption of M13A and the eventual adoption of M13C?
Yes, this is very interesting question. As we are aware, we, due to the capacity of the ICH harmonization, we have to address those various topics in kind of tiers approach, because we do not want to delay the harmonization process to deliver some products earlier than the other. But as we know, these topics in totality related to IRBE is covered probably in multiple regions in one guidance. So there will be some interim period. We harmonize some of the topics, but not other topics. So for the other topics that have not been harmonized, so it will be subject to individual regulatory agencies' regulation. So they should really follow up with the local, like a local region regulatory agencies for input as needed, or follow their current regulatory recommendations. However, as we also heard, just like what we comment on the FDA approach, you can always, if you want, you can always propose some different alternative approach, different from the guidance recommendation and with justification. So we will ask the applicant to use a scientific approach to propose their proposed approach they like to take for their drug development program. Thank you.
Thank you for responding to that group of questions. Moving to our next panelist, we've got a couple more questions that came in for Dr. Nilufer Tampal. And here's the first question. Why in the BE fasting and BE fed states is it not sufficient for drugs with pH-dependent solubility? Why is an additional study with PPI recommended?
Yeah, Ray, thank you for that question. This topic, this is like a newly introduced topic. It's not also covered in the FDA guidance as well as many other guidances of other regulatory agencies. And we have more recently started to focus on this issue. So maybe I'll take a little time to explain why what our thinking is and what our discussions were. So we know that in a sustained elevated pH environment, the absorption and the bioavailability profiles of certain drugs, which have pH sensitivity solubility, are going to be altered. And this is especially true for low solubility drug substances belonging to the BCS2 and 4 class, which are also weak bases, right? And then in presence of the PPI, we know that the gastric pH is going to be elevated. The buffering capacity is going to be reduced. And all this is drastically going to impact the solubility and the dissolution, which will be reduced of the drug substance. And this in turn is going to affect the absorption of the drug. So that is that is why we are going to be testing for the bioequivalence in presence of PPI. Now, this risk, we already do a BE study under fasting and fed condition, but this risk in presence of the PPI will still not be addressed because, again, the dissolution and the absorption profiles may be different compared to the normal pH of a fasting and fed condition. And also, like in the gastric environment, there are multiple processes going on in the fed state. We know that there is change in the volume of the gastric content, the gastric emptying, increased bile salts, many processes that go on. So all these physiological properties of the GI tract can underestimate the impact of the sustained increase in the gastric pH on the drug dissolution and therefore the absorption. So, and this is also apparent to us from the innovators' drug development programs for the different drugs that we see, where their product may be bioequivalent under the fasting and fed conditions, but in presence of the PPI, it doesn't show the bioequivalence, and that's why we have to be testing with an additional study in presence of the PPI. Thank you.
Thank you for responding to that question. We've got a few more questions for Dr. Tampal, and here's the next question. Can you provide the context for how the exclusion of data in exceptional cases relates to the reliability of dose administration?
Yes. Sure. Yeah. Thank you for this question. Yeah. Another good question. So I'll start with FDA's current practice. Right now, our current practice is that outliers may not be removed from based on some statistical test, right? Data, we recommend that the data can only be removed from statistical analysis based on a protocol, not a violation, but a deviation, and that too, which has to be contemporaneously documented. And M13A also includes a similar recommendation for the exclusion of the data. But it makes an exception over here in the M13A. And that exception is that the data may be allowed to be removed due to low exposure, meaning when the subject concentration is very low, such that the AUC of that particular subject in a period will be less than 5% of the AUC of the product in question. So that's how M13A has defined very low concentrations. However, this situation will, in general, can only be applied to no more than one subject in each study. So then this low concentration, it suggests that the subject was non-compliant during the drug administration. He or she somehow managed to avoid swallowing the drug despite the mouth check, which the sponsor claims is done. And when we have recurring instances of this type at a site, it would bring the reliability of the dose administration process and practice into question. So allowing the exclusion of one subject per study, it should not be taken to be a free pass, meaning for every study, you can just drop one subject. The site would need to do its due diligence with respect to the mouth check as well as the dose administration. Otherwise, as I said, the reliability of the processes at the site would be in question. Thank you.
Thank you for responding to that question. One final question that was directed to Dr. Tampal is the following. If studies under both fasting and fed conditions are required according to section 2.1.5, would an additional study on pH dependency still be required? And if yet, what conditions should be selected for the study or studies with the PPI co-administration?
Yeah, Ray, thank you again for that question. So here, I just answered like whether an additional study would be required, right? The answer is yes, an additional study with the PPI would be required in certain cases. But the additional part of the question is what type of meal. And I think for as I said earlier, many of the agencies do not have as much knowledge and experience on this topic at this time. And so that's the reason why details for the bioequivalence study design, as well as the statistical analysis, they are not specified in the M13A, this section of the M13A. However, our plan is that when the guideline is finalized, which would be at step 4, the expert working group may likely develop a Q&A type of document. And the meal type and other specifics of the study design and other things related to this topic, we may include that in the Q&A. So at this time, I don't think I can comment upon what the meal type would be for this type of BE studies. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we have a question for Dr. Stella Grosser. And here is the question. Is the estimand framework ICH E9R1 applicable to M13A?
Hi, thank you for that question. I guess in general, the estimating framework is meant as a way, in my opinion, of clarifying and communicating clinical trial plans. It's not explicitly a part of M13A, and I think the sort of thinking that goes into writing up an estimand, discussing what has to be estimated, what population you're estimating, what you're looking for in your study, that sort of framework is valuable as a way of expressing clearly your plan and your protocol and in your statistical analysis plan. I think the attention to missing data that's part of the estimand and the sort of missing data that's part of ICH E9R1 can be very valuable in longer term parallel design PK studies. And probably we will think about including it in later parts of ICH M13, but at this point, it's not explicitly there.
Thank you for responding to that question. We have a unique opportunity to follow up on another question we asked earlier by Dr. Partha Roy, and I'm going to reread the question. Until the time that ICH M13 is formally adopted, will there be any flexibility in not conducting fed studies for products that are low risk if justification for the same is provided by the applicant?
Yeah, so thank you, Ray. This is a good question. I wanted to kind of re-emphasize also some of the things, some of the thinking around this, right? I mean, the FDA has a long-standing approach or a policy to say that we would not usually ask for duplicative study or a burdensome study, which ultimately does not inform our decision making, right? So that's our long-standing approach and policy. Sometimes it may not look like that, but that's what we always strive for, that we would not be asking for studies which ultimately would expose, as you said, healthy subjects or create other kind of burden that is not needed. So following that principle, I mean, definitely, I think, if there is a low risk product and you have a good scientific justification, then definitely we will encourage you to submit that information. Now, you may not be 100% sure whether that will be applicable in your particular case. You can always come to us with a question in the pre-NDA program, in the pre-NDA meetings, or even in the control correspondence. You can always ask that question based on the information that you have on your product and the reference product that you are referencing. So based on all that information, we probably can provide you or guide you properly on that. But so the answer to the question, again, I think I will reiterate, which Nilufer also mentioned, is that if it, obviously when it is finally formally adapted, until that point, a scientific and regulatory justification can always be made in any of the programs that you are pursuing. So, but thank you for the opportunity.
All right, looks like that's all the questions we have at this time, and let's give a great, huge thank you to our presenters and panelists for their very informative and timely talk, and also for responding to numerous questions that came in. We do have just a few closing reminders. This activity is eligible for an attendance certificate, which supports education, continuing education by SOCR, RAPS, SQA, and ACRP. please refer to our website at fda.gov/CEDAR-SBIA for more details. As a reminder, you will only be able to get the attendance certificate upon completion of the survey, and this remains open for 2 weeks. Remember, that's May the 16th. Please remember to download the certificate of attendance immediately upon completing the survey. It cannot be retrieved after the fact. Let us know your comments and feedback via the survey. This will help guide the design of our future events. Additional tutorials are available through fda.gov/CEDARLearn. On behalf of CEDAR Small Business and Industry Assistance, we hope that you enjoyed today's presentation and found it helpful. We look forward to your presence at future webinars, which will be advertised on our website at fda.gov/CEDAR-SBIA. Thank you for attending, and this concludes today's webinar.